Bismillahirrahmanirrahim. Assalamu alaikum. Student, today we have a new topic of chromosome and DNA, and that is known as genetic code. What is genetic code? So, genetic code is the information which is present in DNA or RNA for protein synthesis is known as genetic code. Let's suppose this is gene and messenger RNA. We know that gene consists of nucleotide: adenine, guanine, cytosine, thymine, adenine, guanine. This is thymine to adenine, guanine to cytosine, cytosine to guanine, thymine to adenine, adenine to thymine, and guanine to cytosine. This gene consists of nucleotide sequence, but these nucleotides carry information for the protein synthesis, but this information in the gene is present in the form of nucleotide, and these nucleotides act as a code, and this code is known as genetic code. But when transcription takes place, after transcription of this gene, the information which is present in this gene is now present in the form of messenger RNA. But this messenger RNA also carry a sequence of nucleotide, and this sequence of nucleotide also carry information for the sequence of amino acid, and this information which is present in messenger RNA act as a code, and this is also known as genetic code. So either the sequence of nucleotide present in DNA, gene, or messenger RNA act as a code, and this code is known as genetic code. Similarly, how formation of genetic code takes place. Genetic code formation consists of nucleotide. What type of nucleotide? In the case of messenger RNA, adenine, guanine, cytosine, uracil, because in messenger RNA thymine is replaced by uracil. So genetic code consists of 4 nucleotides. It means that genetic code is composed of 4 nucleotides, but at a time one genetic code consists of 3 nucleotides. One genetic code consists of 3 nucleotides. Let's suppose we have a genetic code: adenine, uracil, and guanine. Adenine, uracil, and guanine. This is a single genetic code, and it is made of 3 nucleotides. Point to be noted. This is a single genetic code, but this single genetic code is made of 3 nucleotides: adenine, uracil, and guanine. Similarly, we have total 64 genetic code. Out of 64 genetic code, some codes are antisense codon, while some are sense codon. Antisense codon are those genetic codon which do not carry any information for the sequence of amino acid, while sense codon are those genetic code which carry information for the sequence of amino acid. Similarly, these 64 genetic codes carry information for the amino acid, for the sequence of amino acid. But how many amino acids are under the control of these 64 genetic codes? 20 amino acids are responsible, are under the control of Q64 genetic code. But we have some antisense codon. It means that they will not carry information for the sequence of amino acid. And these antisense codon are also known as stop codon. So what are those stop codon? They are UAG, uracil adenine guanine, UGA, uracil guanine adenine, or UAA. These are antisense codon. They are stop codon. While sense codon are those codon which carry information for the sequence of amino acid, and they are also known as start codon. Let's suppose we have a start codon, that is A. Let me clear. Start codon is that sense codon which is present at the position number first of messenger RNA. Start codon will always present at the position number first of messenger RNA. So start codon also act as a sense codon, but the genetic code which comes after the sense codon will which comes after the start codon will be known as sense codon. Similarly, in the case of AUG, also a sense codon. So this carry information for the sequence of methionine. Clear? Acha. In the case characteristics of genetic code, codes degeneracy. Code degeneracy means single amino acid can be encoded by more than one genetic code, but a particular genetic code only specify single type of amino acid. Let me clear. Code degeneration means, let's suppose we have a genetic code AUG. This is responsible for methionine sequence. Similarly, we have another type of genetic codes, that is CUU or CUC. This genetic code is responsible for the sequence of leucine. This genetic code only specifies methionine. This genetic code only specifies leucine. This will only specify leucine. It means this will not specify any amino acid other than the leucine. It will only specify leucine amino acid, not any amino acid. Let's suppose this may be methionine, serine, valine, etc. So code degeneration means in the case one amino acid can be specified by one genetic code. Code degeneration means amino acid can be encoded by more than one genetic code, but one genetic code cannot encode more than one amino acid. Similarly, second characteristics of genetic code, that is code universality. Codes are universal. Let's suppose we have a genetic code that is AGA. This code is present in bacteria and human also, but in both organisms this specified amino acid that is known as arginine. But codes are same in the case of nuclear DNA genetic codes. Mitochondrial DNA genetic code is different from the codes of nuclear DNA. Similarly, the third characteristics of genetic code: no gap is present between genetic code. Let's suppose a genetic code AUG, CUU, AGA. So there is no gap present between genetic code. One code is present after another genetic code. Similarly, the fourth characteristics of genetic code, that is non-overlapping of genetic code. It means we have genetic code AUG, CUU, AGA. This cannot be read as AUG, then the next genetic code UGC or GCU. So this is totally wrong. This can be read as AUG, one genetic code, CUU, another genetic code, and AGA, another genetic code. So each nucleotide consists of separate genetic code. So what are the characteristics of genetic code? Code degeneracy, universality, no gap, and non-overlapping of genetic code. So this is all about genetic code.